Hello, everyone. Today we're going to do an ANOVA analysis in Excel. And an ANOVA, or analysis of variance, is going to look for differences between means in more than two groups. So we have a data set here that's some sample data from an experiment that we used to run live in class, where we have corn plants and we have two strains: dwarf corn that grows to a short height and normal corn that grows to a normal height. And we want to see the effect of a gibberellin treatment, which is a plant hormone, coded GB here, on the dwarf corn and on the normal corn. So we have a control for each of those treatments that is a DI water treatment. So we soaked the corn seeds in these treatments and then let them grow for 2 weeks and measured their heights. So we have the heights of each of the plants. As you can see, the dwarf DI water treatments, they're a couple centimeters. The normal corn heights are up to over 10 centimeters. So we have a pretty good data set, sample size between 18 and scrolling down to 50. Well, it's minus 2 rows, so 51. So there's a few steps to doing our analysis of variance. The actual ANOVA, generating the ANOVA table in Excel, is easy, but there's a few things that we need to do as well as that analysis. So there's 3 assumptions for analysis of variance. We want to check all those. One of them is that the data are normally distributed. So we're going to check that using a histogram. And we've done a histogram a few times. So I've already got the histogram finished over here. Notice that I made a column of data, and I just took and copied and pasted each of the columns of data from all 4 of the treatments out into a single column, so I could highlight that one column of data that Excel wants to make a histogram. So remember, you need the data analysis tool pack to do your histogram. And you want to make all of the corrections that you should be starting to get under your belt by now. All of the digits, the more, getting the gaps, right? Now you might notice that we have this bimodal distribution for our histogram. So one of the assumptions for doing an analysis of variance is the data are normally distributed. But if we have treatments that actually differ from one another, we may see a different size of those measurements. So it's a little bit of an art to try and think about, well, if my means really are different, yes, I could see a histogram that has some different lumps in it. So we're also going to look at the actual means and the standard deviations to give us some insight into what our data set looks like. So we did do a line graph with means and error bars earlier in the semester, but let's review. So we want to put into our little data table here that we have each of the groups, and we want to calculate the means. So I looked at the data set over here, and we have for each of our columns, we have data starting in the 3rd cell. So we can do average A3. Now the biggest or the longest set of data goes all the way down to row 53. We don't have that many samples for all of our columns, but if there's nothing in those cells, then you can use the same formula all the way across, right? You could also do average and then highlight the cells in each of the columns. We want to do the same thing for standard deviation equals STDEV.S. And we're going to do the same cell addresses A3 to A53. If we do that, we've captured all of the cells that there could be data in for the whole data set. And we can just copy and paste that formula across. All right, so we want to just highlight the means and the titles in order to make our line graph. And go to insert. All right, so if we highlight both of the title cells in our little table, we're going to get a, excuse me, we're going to get those labeled across the bottom. So now we have to fix a couple things on our graph, and we're going to add a chart element to do horizontal axis, which is corn, type, and treatment. And we need to add an axis for the vertical, which is going to be length, height. All right, so we've got our data series on here. And what we want to do with the standard deviations is add another chart element. And we want to add error bars. We're going to go to more error bar options and scroll down and click on custom, specify value. And then this dialog box pops up where we want to delete this equals 1 in brackets, and we want to highlight the 4 cells that have our standard deviations already calculated in them. We're going to do the same thing for both the positive and negative error bar boxes and click OK. All right, so this line connecting our different dots together, we're going to switch to no line. And for our markers, we're going to do solid fill and black. All right, oops. So they have that border on them that is still blue. I want to change that to black as well. Sorry, it looked like I didn't have it highlighted, so it didn't do that for me. I need to do it again. Solid line and black. All right, that looks good. Delete that title. All right, so we have what looks like the regular dwarf corn plants. They have a pretty small height. The corns treated with gibberellin grew taller. In fact, it looks like they grew as tall as the normal corn. Normal corns treated with gibberellin look like they grew a little taller than the normal corns, right? But what we want to see, our analysis of variance is going to tell you, given these error bars, right, squinting at the data, what we might notice is that these 3 means are sort of grouped together. There's kind of a big standard deviations, so they may overlap, and that's why we need to do the analysis of variance in order to see. We're probably going to find that this, the dwarf corn treated with DI water has a small mean compared to the other ones. We should find it significantly different just from looking at this graph. All right, so now when we look at our graph and we see, well, we do have some difference between our means, and we go back and we look at the histogram, that we have this sort of normal distribution of this larger size plants, and we also have a normal distribution of the smaller size plants, and that we can live with that because it looks like there's each of the groups potentially has a normal distribution kind of by itself. All right. So our analysis that we're going to do, analysis of variance for ANOVA, is a one-way ANOVA. That means we only have one factor. And the factor is how many, what categories we're putting our individuals into. So we want to go to our data tab and our data analysis tool pack, and we're going to go ANOVA single factor, which is the same as a one-way ANOVA. Input range, I need to scroll back to the top, and I'm just going to highlight the data. Oops. And you can keep scrolling down until you get a rectangle that has all of the data points in it. It's okay if these other cells are blank, just don't type zeros in them. If they're blank, it just ignores them. All right, our output range, we need to pick a place for it to put a fairly large data table. So we're gonna go back up to the middle here and give ourselves plenty of space. All right. I guess I didn't give it quite enough space. I'm actually going to go over here. All right, so the results of the ANOVA actually gives you what your sample size is, your average, your variance. If you didn't want to calculate those first, you could use this summary table to summarize for you. And then we're going to look at the actual ANOVA table, which is the the main result. All right, so we have our source of variation between groups versus within groups and the total. We have sum of squares between, within, and total. We have degrees of freedom between, within, and total. Now this is the sum of the squares and the mean squares are the things that if Excel does it for you, it's really nice. Right, and then it's going to calculate the F ratio and then find the p-value for it using the degrees of freedom from the table and the F distribution. So remember that when we have this notation 1.451 e minus 10, that means 1.45 times 10 to the minus 10. So that's a very small p-value. All right, what happens when we reject the null hypothesis for ANOVA is we know that there is a significant difference between at least two of the means, right? Our null hypothesis is that all the means are equal, and we don't know which means are equal though, okay? So what we're gonna have to do is take all of our pairs of treatments and do 2 independent samples t-tests on them so that we can then answer the question, okay, which of our groups are different from one another? This is a little tedious, but it gives you some practice doing 2 independent samples t-tests. We haven't done those for a few weeks. So if you find a significant result, if you reject the null hypothesis for your overall ANOVA, you need to do follow-up t-tests. But if you don't reject the null hypothesis of your ANOVA and you're saying, oh, they're all the same as one another, you don't need to do those follow-up t-tests because they're all going to say that there's no difference between the means. All right, so having done this many, many times, what I find is convenient to do is that I'm going to open a new workbook worksheet, a new sheet, and I have to do all the pairwise comparisons. I have to do dwarf DI compared to dwarf GB, dwarf DI to normal DI, dwarf DI to normal GB, dwarf GB with normal DI, dwarf GB with normal GB, and normal DI with normal GB. Whew, right? So to make sure I get all those done, I like to copy and paste my data so that all the pairwise comparisons are all spread out in front of me. I see all the ones I have to do. And then I'm just going to go to my data analysis toolpack. I'm going to do t-test, 2 sample, assuming unequal variances. And I'm going to do my variable 1 range is my data in the first group. My variable 2 range is my data in the second group. My output range, I'm going to put it at the bottom. Way down here. That way they can all go in a row. And my hypothesized mean difference is 0. All right, so our interpretation of these results, remember that we're going to look at the p-value for a two-tailed test. We're looking for differences between means, and we have found our p-value here. All right, remember when you're judging whether the p is less than the alpha, when you're doing follow-up t-tests for an ANOVA, because we're doing multiple tests, in this case, we're actually going to do 1, 2, 3, 4, 5, 6 different tests. We're going to take our alpha level that we started with, 0.05, and we're going to divide it by 6. That's going to give us the alpha level we have to match or less than in order to have a significant result for our follow-up t-test. We have to be more strict on those follow-up t-tests than we are on our initial overall ANOVA. Right, I'm going to pause the video here while I go through the rest of the t-tests, and then I'll start it up again so that you can make sure that you did them all correctly. Okay, so I finished up all of the t-tests. It was a little tedious, but it took me maybe, I don't know, 5-10 minutes. So if you want to double check that you got the right p-values for all your t-tests, they're just here left to right, same as you have them in the spreadsheet that you downloaded from Canvas. All right, so you want to scroll back to the top, make sure you're connecting the correct comparison test to the right levels of treatment. And then for each of those pairwise comparisons, you're going to interpret the results just the same. Do you reject the null? Do you not reject the null? Right? Your overall conclusion, you want to go back to the original research question. Is there a difference between normal corn and dwarf corn? Is there a difference between dwarf corn treated with gibberellin and normal corn treated with gibberellin? So we can kind of ask, what's the effect of gibberellin on plant growth across these two different types of plants, right? It's a little tedious, but just make sure that you do all those comparisons, write down all your results, and explain the overall pattern at the end.